When we usually talk about evolution, the mental image is almost always exactly the same for most of us.
Oh, yeah. Fossils in a museum.
Right, fossils, or maybe those sketches of finches with the different sized beaks. We tend to think about the big picture. How a fish eventually becomes a lizard or how a dinosaur turns into a bird.
Yeah, the macro view, comparative anatomy.
Exactly.
Yeah.
But today, for this deep dive, we are doing the exact opposite. We are zooming in, way, way in.
Down to the microscopic level.
Right, because we're going to look at a very specific, and I should say controversial argument. One that suggests if you actually pop the hood on life, if you go all the way down to the molecular level, the standard Darwinian explanation it hits a wall.
And not just a tiny speed bump, a very specific, very complicated wall. We're talking about biochemistry, molecular machines, and a concept that has really stirred up the scientific community. It's called irreducible complexity.
Irreducible complexity.
Ever.
Yeah, and our guide for this deep dive is the biochemist Michael Behe. We've got his book, Darwin's Black Box, as our main source today.
Yeah, and we also have a pretty thick stack of critiques and counter-arguments from other scientists to keep things perfectly balanced.
Which is important. But before we get to the controversy itself, help me set the stage here. Why does Behe think everyone has been missing the point for, 150 years?
Well, to really understand Behe's beef with the status quo, you have to look at what scientists used to think a cell actually was.
Okay.
If you go back to Charles Darwin's time, so mid-19th century, and you looked at a cell through the absolute best microscope they had back then, what did you see?
Well, the notes say it looked like a glob of protoplasm.
Exactly. A simple blob. Maybe you'd see a dark spot in the middle for the nucleus, looking kind of a piece of fruit suspended in Jello. They called it a black box.
A black box, like on an airplane.
Sort of, but in science, a black box is a system where you can see the input and you can see the output, but the internal mechanics are just a total mystery. Darwin and his contemporaries, they just assumed the inside of that box was simple.
Which makes sense, honestly. I mean, if you think the cell is literally just a blob of jelly, then evolving from one blob to a slightly different blob doesn't seem like a huge, impossible leap.
Precisely. But fast forward to today, that view has changed strictly because our technology has changed. Bruce Alberts, he's the president of the National Academy of Sciences. He has this great quote in our research. He says, "We have massively underestimated the cell." He doesn't call it a blob at all. He calls it a factory.
And he doesn't mean that metaphorically, right?
No, he means it quite literally. He describes the cell as an elaborate network of interlocking assembly lines. He says it has artificial languages, decoding systems, memory banks for information storage.
Wow.
Yeah, and automated transport systems. It's incredible.
And unlike a human factory, it can replicate its entire structure in just a few hours. I don't know any car plant that can just spawn a perfect copy of itself by lunchtime.
Right. It's a high-tech world that makes our absolute best human engineering look totally clumsy. And this is really where Michael Behe enters the picture. He's a professor of biochemistry at Lehigh University.
And I have to say, reading his profile, he doesn't strike me as some wild firebrand trying to tear down science.
Not at all.
I mean, he drives a minivan, he's got 8 kids, he seems like a pretty standard suburban dad.
He is, and the funny thing is, he wasn't trying to pick a fight. He actually started out perfectly fine with the standard evolutionary model. He just assumed that God set up the laws of the universe and then let natural selection do all the heavy lifting.
So what changed his mind?
Well, he read a book called Evolution, A Theory in Crisis by Michael Denton. And it made him realize something that he found really disturbing. He kept hearing scientists say things like, "Isn't it wonderful how natural selection created this amazing biological structure?" But nobody was actually explaining how it happened at the molecular level.
So they were sort of skipping the blueprint.
Yeah, they were skipping the nuts and bolts entirely. Behe realized that life is, at its core, a molecular phenomenon. If Darwin's theory is going to work, it has to work down at the level of atoms and proteins.
Right.
And when Behe actually looked at the machinery at that level, he found what he calls irreducible complexity.
Okay, there is the buzzword for the deep dive.
Yeah.
Irreducible complexity. It sounds like a bit of a mouthful. Can you break that down for us?
It is a mouthful, but the concept itself is surprisingly intuitive. He uses a standard hardware store mousetrap to explain it.
Okay, I'm picturing it. The classic little wooden board with the metal snapper.
Right. So think about the parts of that track. You've got the wooden base. You've got the hammer, which is the part that actually crushes the mouse.
Morbid, but yes.
You have the spring to provide the power. You have the catch to hold the hammer back and the metal bar to release it. So 5 parts.
Got it. 5 parts.
Now, here is Behe's test. If you take that mousetrap and you remove just the spring, do you have a mousetrap that works half as well?
No. You just have a piece of wood with some junk on it. It catches exactly 0 mice.
Exactly. It is not 50% efficient. It is totally broken. The function of catching a mouse only appears when all the parts are assembled and working together. That is irreducible complexity. You can't reduce it any further without losing the function completely.
Okay, I get the mechanical logic there. But why is that a specific problem for evolution? Because Darwin talked about small, gradual changes over time. Why couldn't you just add the spring later?
Because of how natural selection actually works. Darwin himself wrote in Origin of Species, and this is a famous quote, that if any organ could be shown to exist that couldn't be formed by numerous successive slight modifications, his theory would absolutely break down.
Oh, wow. So he basically gave us the exact criteria to prove him wrong.
He did. And Behe argues that natural selection operates strictly on the now. It selects for things that offer a survival advantage today. It can't look into the future and say, hey, let's keep this completely useless metal bar because in a million years it'll be part of an awesome mousetrap.
Right, so if you have a wood base and a hammer, but no spring, it doesn't catch mice. It offers 0 survival advantage.
Which means natural selection wouldn't preserve it. It would weed it out. The system has to be essentially complete before it works at all.
Okay, fair enough on the mousetrap. But that is a human invention. Biology is messy and organic. Do we actually see these all-or-nothing machines inside our own bodies?
Behe argues that they are literally everywhere. But he points to the bacterial flagellum as his absolute flagship example.
The flagellum. I remember this from high school biology class. It's the little tail that bacteria use to swim.
Yeah, but calling it a tail really sells it short. It is quite literally an outboard motor. Yeah, it's a rotary propeller, and the stats on this thing are honestly mind-bending for something microscopic. It spins at 10,000 revolutions per minute.
Wait, 10,000 RPM? That's like Formula One car territory.
It's faster. And get this, it can stop within a quarter of a turn and instantly reverse direction to spin 10,000 RPM the other way.
That is insane.
Howard Berg from Harvard calls it the most efficient motor in the universe.
What on earth is powering it? Obviously, it's not burning gasoline.
No, and this is where the biochemistry gets really cool. It runs on a flow of acid. It uses what's called a proton motive force. Basically, the cell pumps protons out, and as they flow back in across the bacterial membrane, they drive the rotation of the motor.
That sounds incredibly high-tech for a common germ.
It is, and structurally, it is a dead ringer for a human-engineered motor. Scientists actually give the parts standard engineering names. It has a stator, a rotor, a drive shaft, a U-joint, and a propeller. And it takes about 30 to 40 different proteins just to build it. They even have specialized constructor proteins whose only job is to assemble the flagellum.
And so I assume Behe's argument is exactly the same here as the mousetrap.
Yeah.
If you pull out the rotor, the bacteria doesn't just swim a little slower.
It doesn't swim at all. It is dead in the water. So the question becomes, how do you evolve a rotor and a stator and a drive shaft all simultaneously? If you just evolve a rotor by itself, it's dead weight. It's a waste of the cell's energy.
And natural selection should weed it out,
Yeah,
not preserve it.
exactly.
It does seem like a massive leap of luck to get all 40 parts together at once. But is the flagellum just a weird one-off? Or is this common?
Behe says it's a clear pattern. The sources also detailed the cilium.
The cilium.
Yeah, these are those little whip-like hairs on the surface of cells, like the ones in your respiratory tract that sweep mucus out of your lung, or the ones that make sperm cells swim. Under the old light microscopes, they just look like simple little hairs.
Another simple blob situation.
Exactly. But under an electron microscope, it reveals a complex machine made of about 200 different protein parts. It is basically an automated rowing machine.
How does a microscopic hair row?
It's very clever. You have these long rods called microtubules, and you have a motor protein called dynin. The motor grabs a rod and pushes it. But here is the catch. If that was all you had, the rods would just slide past each other until the whole structure just fell apart.
Okay, so just having a motor and a rod isn't enough.
Right. So you need a third component called Nexen linkers. Think of them as ropes tying the rods together. Now, when the motor pushes, the rods can't slide apart because they are tied down. So instead of sliding, they bend.
Oh, I see. And that bending is the actual rowing motion.
Yes. So it's a strict 3-part requirement. Motors, rods, linkers. Remove any one of those, and the whole system fails. Behe argues you need all 3 present simultaneously for any movement to exist at all.
Talking about motors and rowing machines is one thing. It's mechanical. But the sources mention something that feels even more impossible to coordinate, which is the logistics of the cell.
Oh, the intracellular transport.
Right, because a factory isn't just about having machines. It's about moving stuff around efficiently.
This is maybe my favorite part of the argument because it is just so relatable. The cell is not a bag of soup.
Yeah.
It is divided into very specific compartments or rooms. You have the nucleus, the mitochondria, the Golgi apparatus.
Right.
Now, the cell manufactures new proteins in a factory called the ribosome. But those new proteins have to get to the correct room to do their jobs.
So how do they get there? They don't just float through the soup until they bump into the right place.
No, floating is way too slow and totally random. They are actually loaded onto molecular trucks.
Trucks?
Yes, enclosed carriers with little motors that actually walk along molecular highways.
You're telling me there are physical highways inside a single cell?
Extensive ones. And here is where the irreducible part really kicks in. The protein needs a ticket, a chemical signal attached to it so it gets loaded onto the right truck.
Okay.
The truck then needs to recognize the destination signals on the highway. And then he calls this the gangplank problem. Once the truck gets to the destination, let's say the nucleus, there has to be a mechanism to actually open the wall and let the cargo inside.
It's literally a postal service. You need the address, the delivery truck, and the key to the specific mailbox.
Precisely. If you have the truck but you don't have the ticket system, the protein gets completely lost.
Right.
If you have the truck and the ticket but no gangplank to open the door, the delivery just bounces off the wall. The whole logistical network needs to be in place for any of it to work.
It is staggering to think this is happening in every single cell in my body right now while we're talking. But there's one example in the notes that feels even higher stakes than just lost mail. Blood clotting.
Oh, absolutely. This is literally life or death. When you get a cut, you need a clot. But it is a very delicate Goldilocks scenario.
Meaning too little or too much is bad.
Fatal, actually. If the blood clots too easily, or in the wrong place, the blood solidifies. You get a stroke or a heart attack and you die.
Okay.
If it clots too slowly, you bleed to death. It has to be perfect, it has to be local to the cut, and it has to be instant.
So how does the body manage that kind of crazy balance?
It uses what's called a cascade. One protein activates another, which activates another. It's about a 10-step process involving roughly 20 molecular components. It's like an elaborate row of dominoes, but it's totally self-regulating.
And Behe's argument here is what?
How do you evolve that gradually? If you have a half-formed clotting system, you either bleed out or you turn into a solid block of jelly. There is very little room for evolutionary trial and error when the error means immediate death.
Wow. Okay, so we've laid out Behe's case pretty thick here. The motors, the logistics, the clotting. It sounds incredibly convincing when you lay it out like that.
Yeah.
But this is a deep dive, and we absolutely have to look at the other side. Behe has critics, and they aren't exactly lightweights. Surely there's a scientific counter-argument to the mousetrap analogy.
There is, and the critics have some very strong points. One of the main critics in our sources is Kenneth Miller from Brown University, along with John McDonald. Miller attacks the mousetrap analogy directly with what he calls the tie clip objection.
Oh, the tie clip. What does a tie clip have to do with a mousetrap?
Well, Miller says, okay, Behe, if you take the spring off a mousetrap, maybe it doesn't catch mice anymore, but you can still use the wooden base as a perfectly good paperweight.
True.
Or you can use the catch as a fishhook. Or you can use the whole broken trap as a tie clip.
So his point is that the parts could have had different useful jobs before they ever became a mousetrap.
Exactly. In biology, this is called co-option. The argument is that maybe the bacteria used the rotor part as a secretory pump long before it became a spinning motor. It had a different job that was useful, and then evolution later repurposed it.
That actually sounds very plausible. Does Behe have a comeback for that?
He does. But he says that argument works backward, not forward. He says it's easy for us, as intelligent agents, to look at a finished machine, dismantle it, and say, oh, I can use this piece as a paperweight.
Okay.
But the real problem for evolution isn't finding parts. It's the problem of assembly.
How do you mean?
Well, even if a protein was happily acting as a paperweight somewhere else in the cell, how does it know to suddenly team up with 39 other specific proteins to build a brand new motor? Behe argues that simply having the parts lying around doesn't explain where the instructions come from to assemble them into a new complex function.
It's the difference between having a pile of bricks in your yard and having the actual blueprint for a house.
Right. And to illustrate the odds of this assembly happening by random chance, Behe uses the statistical analogy involving a county fair.
Okay, lay it on me.
Imagine you are in a massive dark room with 10,000 strangers. These people represent the proteins in a cell.
Okay.
You are blindfolded. You can't speak.
Sounds like a nightmare scenario.
It really is.
Yeah.
Now, your goal is to find two specific people in this dark room. Let's say you need to find two guys named Lee, and you need to link hands with them to form a very specific triangle.
And I'm just groping around randomly.
Completely randomly. The odds of finding the two Lees are astronomical. But in the cell, Behe says it's even harder because the mutation rate is so low. In the analogy, that means you are only allowed to let go of a person and switch partners once a year.
Oh, man. So I grab a random person, realize it's not Lee, and I have to wait an entire year to try again.
Yes. Behe calculates that for a complex machine requiring 5, 10, or 50 specific connections, the time required effectively becomes infinity. He argues that even if the parts existed somewhere else, even if co-option is absolutely true, the actual assembly by random grouping is statistically impossible.
That brings up the other really common argument I hear, which is gene duplication. The idea that a gene just gets copied by mistake, and then that extra copy is free to mutate and experiment without hurting the main organism.
Right. Behe calls this word picture science.
Word picture.
Yeah, he says it sounds good in words, but it doesn't work in reality. He says, sure, you can duplicate a gene, but if you duplicate the spring in a mousetrap, you don't get a wooden base. You just get a trap with two springs.
So more of the same doesn't equal new complexity.
Exactly. He argues that no lab experiment has ever shown a duplicated gene suddenly springing forth with a completely new, complex function. It's a theoretical possibility, but he says the hard experimental evidence just isn't there.
Speaking of experiments, wasn't there a famous one involving E. coli by Barry Hall? I remember reading that it was supposed to totally disprove Behe.
Yes, this is a key battleground in the sources. Hall knocked out a specific gene in bacteria, basically breaking a complex system. And eventually, the bacteria mutated and fixed the glitch. Critics pointed at this and said, look, evolution fixed it. Behe is wrong.
That does sound like a pretty solid win for evolution.
On the surface, yes, but Behe looked closer at the methodology. First, he pointed out that the bacteria didn't build a new system from scratch. They just patched a broken part of an existing one. But more importantly, Hall intervened.
Intervened how?
He added specific chemicals to the petri dish to keep the bacteria alive while they were broken and mutating.
Ah, so he kept them on life support.
Exactly. Behe argues that without that intelligent intervention, without the scientists acting as a nurse to protect them, the bacteria would have just died immediately in the wild.
Interesting.
So, in a way, Behe argues the experiment actually proved his point. It showed that you need an intelligent agent to guide the process and protect the organism, otherwise it dies before it can ever evolve the fix.
That leads us right into the real philosophy of it all. Because the absolute biggest knock against intelligent design from the scientific community is that it's not real science, because you can't test it. You can't put design in a test tube or falsify it.
And Behe actually flips that criticism completely on its head. He says his theory is very falsifiable.
How so?
He says, simply show me the steps. If you can demonstrate a natural, unintelligent, step-by-step path that actually produces a working bacterial flagellum in a lab, his entire theory collapses. He's putting his chin out and saying, hit me.
He's literally asking for the blueprint.
Yes, and conversely, he argues that Darwinism is actually the one that's hard to falsify. Because whenever they can't explain how a complex machine assembled itself, supporters can always just say, well, we just haven't found the pathway yet.
Right.
Behe calls that an argument from ignorance or evolution of the gaps. You just assume evolution did it, even without the evidence.
It's really fascinating how he views the arrow of progress in science. Because usually we think the more we know, the simpler and less mysterious things become. Like, we used to think lightning was Zeus throwing thunderbolts, and now we know it's just basic electricity. But Behe seems to think the exact opposite is happening in biology.
He uses what he calls the flashlight in the garage analogy.
Okay.
Imagine you are in a dark garage, and you shine a little flashlight on a car engine. You see a shape. You know it's a machine. You might reasonably think maybe simple natural forces built this over time.
But then you turn on the overhead lights.
Right. The lights come on. Suddenly you see the fuel injection system, the computer chips, the complex wiring harness. You don't find simplicity, you find vastly more complexity than you ever imagined.
No.
Behe argues that as we get better microscopes and as we map more genomes, the cell is looking more intensely engineered, not less. The data points toward design purely based on the structural evidence of the machines themselves.
So pulling this all together for our listeners, we've gone from Darwin's black box, the simple blob of Jello, to a hyper-advanced factory floor. We've looked at molecular outboard motors spinning at 10,000 RPM and microscopic delivery trucks that need chemical tickets.
And we've seen that these biological systems are incredibly integrated. Behe's Irreducible Complexity argues strongly that you simply can't build these machines one tiny part at a time because the unfinished versions offer no survival advantage. They just don't work.
And while the critics argue that parts can be reused or repurposed from other jobs, Behe insists that the sheer statistical improbability of assembling them by blind chance makes Darwinism a really poor explanation for the mechanics of life.
It really does come down to a fundamental clash. It's the engineering sophistication we can clearly observe under the microscope versus the random, unguided mechanisms we traditionally credit for creating it.
It reminds me of that quote from the astronomer Alan Sandage we had in our notes. He said, "The world is quite simply too well put together."
It's that profound sense of awe. When you really take a hard look at the hardware of life, it's very hard to shake the feeling that there is a deliberate architecture behind it.
And I think that is the perfect thought to leave you with today. Imagine for a second we sent a probe to Mars, and we dug up a microscopic motor in the red dust. Rotor, stator, drive shaft, the works. We would instantly unanimously conclude that an intelligent engineer had been there. There would be absolutely no debate.
Not at all. We would throw a global parade.
Yet when we find an even more efficient, vastly more complex motor humming away inside our own cells, we hesitate to make that exact same assumption. So is it the science that's stopping us from seeing design, or is it our philosophy?
That truly is the ultimate question.
Thanks for taking this deep dive with us. Keep looking closer, and we will see you next time.